Hello, everyone. So I'll talk about three firsts first. This is my first time in the wonderful city of Ahmedabad. So very hospitable, very welcoming city. I was very happy to be here. Um, and secondly, this is my first time attending Diabetes India meeting in person. So thank you for the invitation, Dr. Sabu and many of the other organizations here. And also, finally, this is the first time I'm presenting the last talk on the last day. Thanks to Dr. Sabu and all of you for lingering in here till now. So the title I was given is by Dr. Sabu and others is Treating Type 1 Diabetes, What Does the Future Hold? What I added to the title is the hope for today and the promise for tomorrow. And hopefully I'll be able to convince you about or at least infect you with the excitement we have at Breakthrough Type 1 Diabetes with what the future holds for treatments for type 1 diabetes. I'm going to jump right in here. I have a brief agenda here. I will not be able to cover the waterfront of what we do at Breakthrough T1D, but I will select some areas that are more treatment relevant for this particular talk here. Our mission has always been to accelerate life-changing breakthroughs, cure, prevent, and treat type 1 diabetes and its complications. And one disclaimer I will tell you is sometimes these lines blur. A treatment can be a preventive treatment, it can also be a curative treatment. It just depends on what stage, which I'll get into in a second, you're intervening, what stage of the pathogenesis you're intervening in. But this has been our mission, and we have been in existence for 55 years now. So it's always been our mission to find this. At Breakthrough T1D, I will show you in a second that we actually cover the entire pipeline. Advancing breakthroughs is primarily research and regulatory pathways and processes. Advocating for progress here includes reimbursement, includes policy change, fundamental policy change here, and then improving everyday life with T1D and powering a movement. And I hope you all will become part of the movement, which I'll come to at the end of my talk. Within Breakthrough T1D, we support the entire pipeline. We have bench strength in R&D, regulatory, health policy, as well as government, congressional advocacy level. But our ultimate goal is a person with type 1 diabetes doing better? The yardstick by which we are measured is a person with type 1 diabetes doing better? Did we move the needle in their life? So our mission can say life-changing breakthroughs. Life-changing can be different in sub-Saharan Africa versus in New York. And have we changed their life? And that's how we try to measure ourselves. This is a seminal finding in the field of type 1 diabetes. This took about 20 years of R&D, of research, many funding organizations. This was led at the time we were called JDRF, and our previous chief scientific officer, whose shoes I try to fill in now, is the first author in this paper. There have been many, many key findings, discoveries, and inventions in type 1 diabetes. But if you ask me the difficult task of rank ordering, which is the topmost one, I always go back to this thing. This is the fundamental of type 1 diabetes. To understand the pathogenesis, who is at risk, genetic risk or non-genetic risk, who is going to develop, how are they going to progress, at what point in time do they become insulin requiring, which is shown here in stages 1 through 4 here, where stage 4 are the 9 or so million people diagnosed and living with type 1 diabetes worldwide. And the number might be off, but around that, and it's about 1.6 million people in the United States of America. At Breakthrough T1D, like in any organization, you have to prioritize. You have to prioritize. And today I will not be able to focus much on disease-modifying therapies, which are mostly, as the word suggests, did you change the biology of the disease, the pathology of the disease? Did you modify the course of the disease, i.e. the autoimmune process and the frank loss of endogenous beta cells or insulin production? I will not have time much to get into this here. I will spend most of my time today on stage 4, i.e. all the people living with frank insulin dependence today or type 1 diabetes here. I'll focus on cellular replacement therapies and treatment paradigms here. First, I know this is a very knowledgeable crowd, and I'm very thankful to the presenters yesterday and today morning and the recent presenter here to talk a little bit about type 1 diabetes here. But largely, there has been a lot of myth, a lot of dogma. So this is kind of a level-setting, myth-busting slide here, which many of you may be familiar with, but I think it's worth a repeat. If you have a familial risk, a first-degree relative, which is parents, siblings, children, you have a heightened risk of 15, sometimes 20-fold heightened risk, depending on how many parents have T1D. That is indisputable. However, 85 to 90% of the people newly diagnosed with type 1 diabetes, 85 to 90, mind you, do not have a known family connection of type 1 diabetes. There has to be something beyond genetics here, because genetics doesn't change that rapidly. Second myth buster, and Dr. Sabu teed it up for me. I would say he stole my thunder a bit. 50% of new diagnosis happens above the age of 20. It varies a little bit between 45 and 55%, depending on the geography you're looking at, and we don't have epidemiological data in every part of the world. But about 1 in 2 people newly diagnosed are above the age of 20. It is no longer a juvenile onset disease. Again, Dr. Sabu teed it up. 40 to 44% of the adult onset type 1 diabetes are misdiagnosed, mostly with type 2 diabetes, sometimes with LADA. And this becomes relevant in my next slide. I talked about the prevalence right now. And we used to think, and we say this in the United States a lot, it's no longer a white man's disease. No longer Caucasian only. The population that has the highest incidence in terms of the, not prevalence, incidence in terms of the rising rate in the United States is Hispanic Americans. Higher than the Caucasian population. So something has to be looked into here, including genetics here. And unfortunately, India has the largest number of people below the age of 20 with type 1 diabetes. I think it's about 900,000. These are important facts to remember as you go about your clinical research or even clinical practice here. Ask the question before diagnosing or not diagnosing with type 1 diabetes just because an adult overweight walks into your clinic. This is the only slide I'm going to show on early detection. It's extremely important. We are currently supporting, everything shown here in blue is currently supported by us. There are many other screening programs here or early detection programs here, trying to identify two things. One is, how can we find these individuals early on that have autoantibody presence, maybe a genetic risk factor, genetic predisposition. How can we identify them both in pediatric and in adult population? Because unless you identify them, you can't intervene. Can't intervene, you can't avoid DKA at diagnosis. So we have several programs. The second goal for us is to generate the evidentiary basis for cost effectiveness of screening so that not just in the United States, but around the world, federal agencies cover the cost. Just like they cover the cost of screening for cholesterol and many other risk factors, which we have heard a lot about this morning. We have a few miles to go before we can say we can declare victory, but we have very strong evidence coming up right now about cost effectiveness in pediatric onset screening, but we are yet to get there for adult onset. This is the only slide I'll show on early detection. I will encourage here, and I was speaking to a couple of the clinicians here earlier today about this screening here and also avoiding the misdiagnosis, particularly in adult onset. I think if you talk to somebody from Barbara Davis Center in Denver, they will say the reverse is also true. An adolescent walking into the clinic with high blood glucose is not necessarily type 1, could be type 2, which I'm sure you all will agree with. So we have to be very, very judicious in our diagnostic criteria and then offer the treatment accordingly. Let me jump into the first of two topics. At Breakthrough T1D, we always use the plural of cures for two reasons. Biologically, I don't think there's going to be a panacea. There's not going to be a one-size-fits-all because this disease is also, like almost all NCDs, this is also a very heterogeneous disease. It has a very different presentation. There have been studies done in identical twins that have presented very differently with their type 1 diabetes. Therefore, I don't think there's going to be a panacea. So biologically, we will need multiple cures. The second reason is that if you look at cures, you can define it as disease-modifying therapy where you address the underlying pathology of autoimmunity and frank loss of endogenous insulin production. Alternately, you can provide insulin independence to the individual where you don't correct the disease, but you provide insulin independence, a huge relief, and reduce long-term complications. That's the topic I'm going to talk about today. Our biggest confidence comes from islet transplantation, where people are 15 years out, 18 years out, both in Canada and other parts of the world, that are now insulin independent from deceased donor islet transplantation. That gave us the MO that if we can replace these, then we will be able to provide insulin independence and a form of cure to these individuals. Here, you have three main scientific challenges that we focus on. Because we know deceased donor islets is not a scalable solution. It's not a scalable solution, and it requires lifelong, chronic, broad immunosuppression. It's not a scalable solution. So the first is cell source. And can we generate stem cell-derived beta cells or islet cells that are faithful, reproducible, and scalable? The second is the survival of these cells. How can these cells survive at the site of implantation? Because I don't think hepatoportal vein infusion, which is what is currently done, is going to be a viable solution. It's a very hostile environment, as you know. 70% of the islets are lost within 72 hours of the implantation. So how do you protect these graft versus host disease, depending on your site of transplantation? So we have a focus area on cell survival here. And we're using a lot of ML AI here to learn from other transplants fields to understand what induces rejection and what promotes acceptance of a graft. Thirdly, and the million-dollar question where most of our funding is going right now, is cell protection. Cell survival is about the transplant site. Cell protection is the cell per se. How do you protect it both from the autoimmune and alloimmune pathways here? Don't forget, you're putting the cells back in the person that has autoimmunity. Even though they may be 30 years out with type 1 diabetes, you may not detect autoantibodies in their serum. But the moment you put beta cells in there, and Minnesota did a very cool study in the 90s or early 2000s, Dr. Bernard Herring, where he put these cells from identical, maybe syngeneic twins. When put these cells into the other twin, immediately the autoimmunity became active. So there's an immune memory, just like you have metabolic memory. So you have to protect against the autoimmunity and, of course, the alloimmunity. So most of our support is going on here. I am very happy that the first problem of cell source has more or less been resolved. We know there are several protocols with slight deviations from each other, which faithfully differentiate stem cell, both embryonic and adult stem cells, from iPSC cells, from stem cell origin to beta cells. And Professor Doug Melton at Harvard was the first one who showed this. We founded him for about 17 years to show this one here. But now several other investigators are able to do this. So I think we have solved the first problem. Our focus in the cell source is manufacturing, so scalability, durability. We don't know what the shelf life of these cells are going to be. For a drug, we know it's 6 months, 1 year, 2 years. But we don't know what's going to be for cells. And then what's the safety? Because the last thing you want to go and say, I cured type 1 diabetes, but guess what? I gave him cancer. So that's another hurdle, is the safety of these. So our focus areas right now are manufacturing, which includes cryopreservation, shipment, scalability. We have to be making hundreds of billions of cells. And right now we are at the 5 billion mark. So Breakthrough T1D is supporting 3 cell factories. That is not doing core research. That's just doing manufacturing. The second problem, protection. There are many ways to protect the cells, and this is just one summary slide of 15 years of intense work by the Breakthrough T1D Beta Cell Consortium. 50 plus investigators, 15 plus companies. Regulators are also involved in this. So one that has been around for a while is encapsulation, where you provide a biomembrane, a semi-permeable biomembrane for nutrient exchange, insulin delivery, but not allow the larger molecules, the immune molecules, the antibodies to infiltrate into the cells. Each of these approaches have advantages and challenges, as you can imagine. Second is local or targeted immune modulation. And I'll give you one example shortly. This includes 3D bioprinting, bioscaffolds, local pro-drug delivery like parathyroid hormone or the anti-CD40 ligand that we reported on in October, and other approaches here to remove the broad immunosuppression, which is deleterious. It's the hammer approach with ATG, tacrolimus, and all of these things. Can you go in with a very selective, a milder immunotherapy, not immunosuppression approach? And finally, the holy grail is make smart beta cells. Gene-edited beta cells that are camouflaged from the body's autoimmune and the alloimmune attack on the cells here. It's quite a ways to get to gene-edited, but Sana Biotechnology has reported something that I will share with you in a second here. So this is where most of our approach is here. Right now is to identify what are the ways we can protect the cells once they go into the body here. There have been significant advancements, which is why we are very excited at Breakthrough T1D. This is kind of a timeline trajectory here. I don't know if you can read the small font, I apologize, but I'll make my slides available if you're interested. I think the organizers will be happy to share them with you. Basically, this is only clinical. This is only clinical, not preclinical here. You all have heard about Vertex. I have one slide coming up on that, but it's not a one-trick pony anymore. We have several companies, small biotechs as well as mid-sized companies now involved, which is really good for our disease. Because in type 1 diabetes, it's been very difficult, other than the insulin and the device space, to get large pharmaceutical companies to be interested in type 1 diabetes, because the ROI is just not there. And so that's there, and if you walk this, I won't belabor this whole slide here. And today, the disease donor islet is regulated in the United States. I know it's not regulated in other parts of the world. And Lentidra was approved in 2023, June of 2023. So almost 2 years back. Guess how many people have been infused with the disease donor islet product Lentidra? Take a guess. 1. In 20 months. It's just been very, very challenging to do this. So stem cell-based is the solution here. There is significant unmet need here. And right now, when you do immunosuppression, it's approved only for severe hypoglycemia or impaired awareness of hypoglycemia people, which is a very small sliver of the T1D adult population. We want to make it available for all people with T1D, and only way to do that is to reduce the immunosuppression. And that's why I showed you the protection slide before. A little bit of results here. You all may have followed Vertex's clinical trial ongoing. They started small. The FDA allows you only to do what we call a sentinel design in cell replacement therapy, 1 or 2 patients at a time. And when you develop some safety, then they allow you to enroll 5 to 6. In any case, really, really good news. I won't get into the details, but almost everyone is showing insulin independence. Now, the time to insulin independence can vary in individuals. That's where the responder, not really non-responder, but super responders and moderate responders come. Some in 6 months, some as early as 4 months, and some 1 year out. But the bottom line is they're becoming insulin independent. Their A1C is below 6. They have 0 hypoglycemia, and they don't have any product-related, they have immunosuppression-related side effects, but no the cell-related side effect, which I think is an excellent profile. And Vertex announced in November that they have now converted their trial into a pivotal trial. FDA has cleared them to make this a pivotal trial. So we're keeping our fingers crossed. This is their first product. Their second product, VX264, not shown here, is actually an encapsulated product, which I just showed you in a previous slide, which is without immunosuppression. And the results will read out at ADA. So if you're attending ADA, Vertex will present results on VX264. As I mentioned to you, this is not a one-trick pony. We are currently funding a lot of these companies here. Let me talk a little bit about Sana. I don't have a slide, but Sana Biotechnology. So this is a small biotech company here. They have a proprietary platform called HIP, Hypoimmune Platform. And they can use this for any cell replacement therapies for any disease condition, like muscular dystrophy, type 1 diabetes, so on and so forth. So end organ damage diseases. So what they did is they used, just for proof of concept, they used disease donor islets, cadaveric islets, and they introduced mutations, a cassette of genetic mutations, which is proprietary, and they put it in the forearm of one adult in Sweden. And the 28 data looked very promising. This is without any immunosuppression. That's why we want to go for the holy grail gene-edited cells here. And Their press release was out in January. They showed that the cells lodged in the forearm. They were functional till 28 days through imaging. C-peptide levels went up because these were C-peptide negative patients, and so on and so forth. I'm hoping they'll report some clinical data on A1C and other things shortly, but I don't know the ETA on that. And then there are other companies. Eledon Pharmaceuticals has a proprietary anti-CD40 monoclonal anti-CD40 ligand monoclonal antibody. They're pursuing this for organ transplantation. Their main program is in kidney transplantation. We reached out to them and used it in a trial in Chicago, and the results were through the roof. So they did a comparator group of regular immunosuppression, broad immunosuppression, so tacrolimus, calcineurin inhibitors, ATG, so on and so forth, and versus anti-CD40 ligand with the anti-inflammatories, of course, TNF alpha and all that. And they not only showed no side effect profile, but also better engraftment and better results here, and less and more survival of the implanted or transplanted islets here. So it's very promising right now. Yes, early days. Phase 1, phase 2. Early days, except Vertex, which is in phase 3 trial. And so on and so forth here. So this is where cell therapies are today, and I'm hoping that there will be, again, I said hope for today, promise for tomorrow. How can we get there? Let me switch gears to the last part of the talk, which I think you all are quite familiar with. There have been quite a few talks. Dr. Vygorsky gave a talk yesterday. Dr. Basu gave a talk this morning, and many others here. I missed a few of the talks, but gave a talk which tees up my section quite well. Actually, last night I went and I removed some slides because I saw that in some of the other talks there. But improving lives is a broad, intentionally we use improving lives and not treatment alone, because that's very limiting, I think, at least at Breakthrough T1D. Because we look at this as improving health and happiness. The mental burden of T1D, 24/7, 365, which I'm sure there are people with T1D in the room can attest for it, is insurmountable. And it's a family disease, as all of you know, those that have it there. So we're looking at the 4 areas of focus are devices, which you all know, closed-loop systems, then adjunctive therapies for both glucometabolic control and long-term complications, which you've heard a lot about in this meeting here. And then also we have a small but meaningful program in psychosocial interventions. And how can we have these with the CBT therapy, mindfulness, how can we roll it out through a clinical trial evidence base so that then the healthcare systems can cover it? It's very, very difficult, at least in the United States, to get coverage for these things, even when you have the evidence. So we are advocating for this as much as we can. I do not need to spend time on this slide. And every time I make this slide, within a month, it's outdated. That's the progress. This field is thriving. This field is thriving thanks to a lot of support at the time JDRF did through our Artificial Pancreas Consortium, started in 2005. And now we have dialed down here, and we are focusing on niche areas where we can be impactful in very basic R&D. For example, transformative is a very broad term. Transformative can be miniaturized devices, inexpensive devices, combining CGM and CKM, and we have 5 projects, 2 with biotech companies and 3 academics. And as you know, Abbott just submitted for CE clearance in Europe for their CGM CKM device. And they will be shortly submitting to FDA, I am anticipating. So we have an effort in that. We are strategic in where we invest, because we don't invest a whole lot in devices now. And then, can you combine a CGM and a pump? Guess what? It's not a technical problem. It's actually a business problem. Because the CGMs are now becoming 14 days, soon to become 28 days. The pumps are 3 to 7 days. It's a huge business challenge. And other than Medtronic, no company has both. So it will take a little bit of a business acumen to get us there. Technically, it's not impossible to combine the two, which is the good news. And then AID systems with more automation, because there are people still miss meal bolusing. People still struggle with exercise, so on and so forth. And then, algorithms, we're supporting ultra-rapid acting insulins. Any ideas? Please reach out to me. Any other ideas? So we don't want to be pigeonholed into what we think is the way of the future. We want to entertain ideas from others, from you and several others around the world here. And in your own backyard here, not here, but in Bangalore, we are supporting a transformative pump. This pump is being developed by Dr. Manish Arora and Deval Karia. at IIS in Bangalore. What they're trying to develop is a pump for low and middle income countries. And we are supporting their development. They've made some very good progress, and hopefully they'll be able to get to the finish line very soon here. And they're trying to develop a novel, affordable pump for initially for people with type 1 diabetes, but eventually, the sky is the limit here. And reduce the cost, because this is a huge burden. I'm preaching to the choir. I don't need to be telling you all about this. Very quickly, let me go to adjunctive therapies here. And I was talking to Dr. Goklani this morning. T1D needs a paradigm shift. We come from a very insulin monotherapy, insulin-centric view of the disease, and we are blinded, we meaning researchers, clinicians, even the people with diabetes community, we are blinded towards opening and looking what doesn't meet the eye. The benefits of adjunctive therapies are humongous here. And why do we need these? There are some stats here. I apologize, these are only US stats here. But I would posit the stats are quite similar outside the US, if not worse. 2/3 of the adult population is overweight or obese, which may not be true in India. 70% are not reaching their goal. Dr. Sabu just asked this or made a comment in the previous session here, are not meeting their ADA goals here. And long-term complications, although significantly reduced since the DCCT edict times, still prevail. And don't forget there's metabolic memory. If you had poor A1C from age 5 to age 25, and then you became a well-controlled T1D, unfortunately your body doesn't forget. There's epigenetic modifications. It comes back to haunt you later. And how can we change that here? Every chronic disease is treated with polypharmacology. Why not T1D? I do not get any money from Novo Nordisk. This is just an example. Just an example here, using Ozempic here. It's an old slide. Basically, the value of these things here. I'm not going to belabor this because there were excellent talks on single and dual GLP, GIP agonists, and there'll be more coming down the pipeline from various companies. Even triagonists and tetraagonists are coming. So changing the enteroendocrine hormone system and trying to rebalance it. The long-term effects are TBD at the moment because these are fairly new drugs. Not the GLP ones, but the dual agonists are fairly new drugs. So it's a little bit of a TBD at the moment. All of these sounds very rosy, very nice. What's the but in the sentence? T1D is an exclusion criteria for all pharma trials. We are trying to fill that gap here. And then I'll skip this slide. Basically, multi-drug approach. We are combining GLP-1 with SGLT. A trial is running between New York and UK, Scotland right now at the moment here. And then, we are trying to look where in US we use this expression because they play a lot of ice hockey. Think where the hockey puck is going to be, not where the hockey puck is today. With that in mind, if you look at long-term complications, I'm only giving the example of kidney here, the KDIGO guidelines. You can say the same thing from American Heart Association guidelines. And you can go on and on. Do you see the blue boxes there? Some have been approved as first-line therapy. What's the catch there? T2D. The guideline, because there is no evidence in T1D. We don't have reason to believe it won't work in T1D, but we don't have the evidence. So we are funding pivotal studies right now in partnership with pharma companies to be able to change that here. And we are talking to the regulators, FDA primarily, to allow us for a pathway because we won't be able to do MACE studies or and ESRD studies in T1D. We just don't have the resources, and we don't have 5,000 people with T1D to be able to enroll in a trial. And these are two examples of the pivotal studies we are funding, one with finerenone and one with a dual SGLT inhibitor. And we are doing the same thing with heart failure and also with GLP-1 agonists. But I don't have time to get into all of these things. My final slide here is where I started. Thinking of type 1 diabetes. I've been hearing the word holistic a lot in this meeting. I won't use that word. Think of type 1 diabetes as a different disease, not just a metabolic disease. Autoimmune disorder that can occur to anyone at any point in their life here. And what has been in the past, we need to be looking forward to what's in the right. And the right-hand side is not an exhaustive list. I just gave a few examples there. How can you help us all get there faster and in a more efficient way? That's the trick of the trade here. Because the disease isn't stopping or waiting. So advanced breakthroughs with us. I'm urging you all, I'm pleading you all to be part of this movement. Do it in your local geographies in India and in partnership with us, and figure out a way where this can become a concerted effort in different parts of the world, particularly in India, where T1D is maybe not as prevalent, but at least the raw numbers become very high when the denominator is high, of 1.4, 1.5 billion people here. Thank you. I always end with this slide here. The two women in the slide are our founding mothers, Lee Ducat and Carol Lurie. Lee Ducat is still alive in her mid-90s, and Carol Lurie passed away a few years ago. In the late 60s, they both had one son each. They had other children too. They had one son each with type 1 diabetes, one in Philadelphia, one in New York. We have no idea how they connected. Pre-cell phone, pre-internet, we have no idea how they connected. They went from door to door, doctor to doctor, researcher to researcher, went to the head of NIH at the time, Lester Salans, and they were appalled that the federal funding and awareness about type 1 diabetes was exactly zero. That's when they founded, at the time we were called JDF, in 1970, in the living room or drawing room of Lee Ducat in Philadelphia. And this is a very inspiring story because our foundational parents have been very instrumental. Of course, today, we are thinking about adult onset T1D also. So the whole thing has changed here. But I thank you for your attention and thank you for lingering to the last minute here.